biologist from Massey University in Auckland. Welcome to the show, Heather.
Good to be here, thanks.
Could you tell us a little bit about what a microbial evolutionary biologist is? It's quite a mouthful.
It is a mouthful. I apologize for that.
It's okay, I managed it.
So it's a lot of things, actually. I have a small research group at Massey University, and what we're interested in doing is studying why bacteria have evolved to be the way that they are. And that's, an incredible thing to get to study, because bacteria have been evolving for billions of years longer than we've even had kind of multicellular organisms on the planet. So we're all kind of like this last minute flash in the pan, and microbes like the bacteria that I study have been evolving and being in contest with one another for much, much longer.
So Heather, what are you going to be drawing for us today?
I think today we're going to talk a little bit about bacteriophages and how we're planning on using them to help honeybees here in New Zealand.
Exciting, can't wait.
So the first thing to say is the microbial world is this amazing and diverse place, right? So bacteria live in every part of the world around us. They live in our guts. They live everywhere. And if you look at them, often they'll have, a little tail so that they can swim. Most are rod shaped, but some are spherical. So that's a little dude kind of in the process of dividing. And they're everywhere. And most of them are completely harmless or doing really great things for us. And that's important because that's not true of all of them. And the ability to selectively target the ones that are bad and not the ones that are doing great things is an important thing, especially in an era where we're trying to figure out what to do about bacteria becoming increasingly resistant to antibiotics. So sometimes when we use antibiotics, they're almost like a nuclear bomb, that if this is the guy who's hurting you, a staphylococcus for example, then you send in the antibiotics and they wipe out huge numbers of organisms. So they hurt these guys who are actually helping us. So that's not great. And so one of the reasons that we've got interested in the viruses that are able to kill bacteria is because they're highly specific. So viruses come in a lot of different shapes. The viruses that we're interested in are often drawn like this. So this is a virus of bacteria, or what's called a bacteriophage, or just phage for short. So they can look like this, they can just look like this, where they don't have a big tail, or they can look like this. These are all different major types of bacteriophages or phages. And for every bacterial cell that we might want to kill, there are estimated to be about 10 bacteriophages that are specifically probably like targeting just that particular bacterial cell. So they're highly specific. If you send in an army of bacteriophages that are able to kill a particular bacterial cell, they'll only kill that bacterial cell. So phages are awesome. We've known about them for a little over 100 years, and they've been used in places like Russia and Georgia and Poland as medicine since before the discovery of antibiotics. And our laboratory is really interested in the idea that we might be able to use bacteriophages to solve problems. And maybe we don't know quite enough today to solve all of the problems of human infection that we might be worried about. But we are at a stage where there is very good evidence that we can use bacteriophages to solve problems caused by bacteria in industry. So a new project that we have in our laboratory is the ABATE project. And this is active bacteriophages for AFB eradication. So what is AFB? AFB is a disease that affects honeybees in the hive. So it actually affects the larvae of honeybees. And this is a disease that we have been trying to eradicate in New Zealand for the last 30 years. There's a plan to eradicate AFB, and one of the things that happens is that if a beekeeper recognizes that their hive has been infected with AFB, then as soon as they recognize this disease, they have to burn the hive, which is awful, because it means that all of the little bees in that hive are destroyed, which is pretty sad for the beekeeper and pretty sad in general. So here's our little bee. All right, so it's been shown in the United States by some work that there are bacteriophages in the soil that are able to kill the organism that causes AFB. And what we're doing now in New Zealand is we've isolated some strains of the AFB causing organism, which I'll say is Penny Bacillus larvae is the bacterium. So this is Penny Bacillus larvae. And right now we have 5 isolates of Penny Bacillus larvae, and we're gathering soil samples from all over New Zealand so that we can find the bacteriophages that are already in our soil that are able to kill this particular bacterium. And the way that we do that is we have people send us soil samples. So if people will send us their address, we'll send them a little envelope that's pre-addressed and pre-paid that has 3 little sample tubes in it, and they can put soil or bee debris or anything that they like in that tube. And what my PhD student will do is take that and grow it with the Penny Bacillus larvae to see if we can find any bacteriophages that will grow. So she grows that over 2 days in a culture. And then once that's grown, if there are any bacteriophages that have grown in that culture, in order to find them, we put that through a very, very fine filter so that the bacteria can't pass through, but the bacteriophages can. And then we put this solution on an agar plate of the bacteria. And if we see the next day regions where the bacteria are dead, that's the sign that we have bacteriophages. So that means we've found a single bacteriophages that's able to kill this. And if anybody in New Zealand sends us a soil sample or bee debris that has a bacteriophage in it, they get to name the bacteriophage. So my undergraduate students at Massey have done projects like this with different organisms, and they've come up with names like Fancy Pants and Robin, which is the name of a chicken. And once we've found a handful of these that are able to kill different versions of these 5 isolates, we're going to put those together in a cocktail and see if they can actually protect beehives so that they are not infected by Penny Bacillus larvae and so that they don't acquire this AFB disease. And so that's the project that we're doing. We have a website, and people can write to us in the contact there, and we're happy to send out envelopes so that anybody can participate in this project.
Could you just sum up for us why we want to study bacteriophages?
Bacteriophages have a huge impact on the microbial world that we're living in. And beyond that, they are fascinating and have really cool ways of evolving. But even beyond that, much of molecular biology and the kinds of amazing things that we're able to do today really sprang from studying bacteriophages in the first place. So our ability to do a lot of the genetic engineering that we're interested in. So they're a simple entity that provides us with this amazing window on the molecular world around us.
Well, thank you so much, Heather, for teaching us about bacteriophages and how important they are for applications like the honeybee industry in New Zealand.
Thanks, Hannah.